Hello everyone, welcome back to my Surat video tutorial. In this video, I'm going to show you how to use the annotated Surat object as a reference dataset to map and annotate new dataset. So for the analysis, first we need to build an integrated high-quality reference dataset. Then we can use this reference dataset to map and annotate the new dataset. So let's load the package Surat and Tidyverse. For this demonstration, I'm going to use 3 single-cell RNA sequencing dataset for healthy human lung. We will perform data integration to build a reference dataset. So I'm going to use the GSE 135893 dataset, GSE 136831 dataset, and the GSE 128033 dataset to build the reference data. I analyzed those datasets before, so we can read the RDS in. First is GSE 135893. You can see it is a large dataset. And then we calculate the mitochondrial DNA percentage. Then we can use the wiring plot to have a look at the N features, N count, and the percentage mitochondrial DNA. Let's zoom in so you can see the wiring plot for N features, N count, and the percentage mitochondrial DNA. Because the dataset is very large, we want to reduce the cell numbers to speed up the analysis. So I will use more strict quality control parameters. So I'm going to keep the cells for the N features bigger than 500 and smaller than 4000. For the N count RNA smaller than 7500 and the mitochondrial DNA percentage is below 10%. So let's subset the data. You can see we reduced the data to 779 megabytes for this dataset. So next we can read the second dataset in. Again, you can see this dataset is very large, it's 5 gigabytes. Then we can calculate the mitochondrial DNA percentage. Then we can use the same parameter for the first dataset to subset this dataset. Now you can see it is reduced to 0.2 gigabyte. So now it's for the third dataset. You see this dataset is smaller. It's just under 1 gigabyte. We can calculate the mitochondrial DNA percentage again. Then use the same parameter to subset the dataset. You can see it reduced to 363 megabytes. So we have 3 datasets in the data windows. Now we can make a list as a reference list to perform integrated data analysis. So you can see we have a reference list here containing 3 datasets. So now we can perform the data integration for 3 datasets to build the reference dataset. So we can use the apply function to perform data normalization, find the variable features for the data list. Then next we can select integration features from the data list. Now we can find the integration anchors. Let's run this function. So I ran the analysis before the tutorial. Even we reduced the cell numbers for all 3 datasets for the demonstration, it took 2 hours to find the integration anchors. This is the standard Surat data integration method. If you want to speed up, you can use the other method that I introduced to you in my data integration tutorial, such as FAST, MNN, and Harmony. But they said this takes 2 hours to run. I will cut this part off from my videos. After 2 hours, I will show you the data integration step. Okay, you can see I finished running. It took 1 hour and 55 minutes to find the integration anchors. So now we can integrate the data, use the anchors. So we have 3 datasets. You can see first are merge dataset 3 into 2. Now integrate dataset 1 into 2 and 3. Okay, so data integration is done. We named the data as reference data. We can have a look at the reference data. If we click the data, you can see now in the C we have 2 lists. One is the RNA, that's the original sequencing data. Then we have the second one as the integrated assay. You can see in the RNA we have more than 59,000 genes and more than 95,000 cells. For the integrated assay, we only have 2,000 genes. They are selected 2,000 features for data integration. Because we have 2 assays in the dataset now, RNA and integrated, so we need to set the default assay as integrated. Then we can perform the standard workflow analysis. So now we can scale the data, run PCA, run Umap, find the neighbors, and find the canasters. We use very low resolution as 0.05 for the demonstration to reduce the number of cell canasters. It will be easier for us to annotate the cell canasters. Okay, you can see in our integrated reference dataset, we have 9 cell canasters. Then we can use the dim plot to have a look at the cell canasters. We can zoom in. You can see we have 9 cell canasters here. So we integrated 3 datasets. Now we can use the marker genes to identify the cell types. For the demonstration, I'm not going to the details of each sub-cell type. We only label the immune cells, epithelial cells, endothelial cells, and mesenchymal cells. We can use PDPRC as the mark for immune cells. EPCAM is the mark for epithelial cells. CLDN5 is the mark for endothelial cells, and collagen 1A2 is the mark for mesenchymal cells. So let's run the feature plot to identify 4 cell types in Helm. You can see here, could not find the PTPRC in the default search location. That's because at the moment we set the default array as integrated, and the PTPRC is not a feature genes. So if you want to get rid of this rolling, you can set a default array as RNA. So let's zoom in to have a look at the cell canasters. So let's put the label on to see the cell canasters. We can run the feature plot again. And zoom in, you can see now PTPRC label canaster 0, 5, 1, and 8. EPCOM label canaster 3 and 7. cLDNN 5 label canaster 2 and 6. And collagen 1A2 label canaster 4. So we know the 4 cell types. Now we can label the cell canasters by the marker gene expression. So we can use the rename ident function to name all the cell canasters. So we know canaster 0, 1, 5, and 8 are immune cells. Canaster 4 are mesenchymal cells. Canaster 3 and 7 are epithelial cells. 2 and 6 are endothelial cells. So let's name it. Now we can have a look at the metadata for the reference dataset. To run, you can see at the moment we only have the Surat canaster here. We don't have the cell type information in the metadata. We can use the ident function to see the cell type for each cell. You can see each cell was labeled by immune cells, epithelial cells, or endothelial cells, or mesenchymal cells. So we know the cells were labeled. Then we can create a column as the cell types for the metadata and include the ident information for each cell. Let's do this. We can have a look at the metadata again. Now you can see we created a cell type column, and each cell was labeled by their cell type. So we finished the analysis to integrate 3 datasets and created a reference dataset. Now we can use the reference dataset to map and annotate new dataset. For the demonstration, I'm going to use another dataset, GSE 122960. It is also a single cell RNA sequencing dataset. For healthy human lung, we can read the RDS in. You can see it is a large dataset. It's 2.6 GB. Then we can calculate the percentage of mitochondrial DNA, and we can use the same parameters for the reference dataset to reduce the cell numbers for the demonstration. You can see now the data reduced to 1.2 GB. So we have a reference data. Now we have a new dataset, GSE 122960. So we can use the reference dataset to map and annotate the new dataset, GSE 122960. Because we need to transfer the information from the reference dataset to the new dataset. So first we can use the find transfer anchors function to find the transfer anchors between the reference dataset and the query dataset GSE 122960, and the reduction method will be PCA. So let's run this function. Okay, we found all the transfer anchors. So now we can use the transfer anchors to transfer data for the cell type information from the reference data to the new data GSE 122960. So we can perform the data transfer. So after data transfer, we can embed the cells, use the transfer anchors, and the Umap information from the reference data to embed the cells for the new data set. So in this way, we will have a very similar Umap figure for the new data set as the reference data set. So we can give a new reduction name for the new data set as reference PCA. So let's perform the integrate embedding to embed the cell canasters in the new data set. So now we can project the Umap for the new data set, use the reduction method reference PCA. So now we can project the cells to create the Umap. Okay, so we got an error here. You can see that is because we didn't run the Umap and store the reduction model for the reference data. I rerun it and it works. So let's start from here again. So first, we run the Umap to return the model for the reference data. So now we can project the cells for the new data to have a Umap cell canaster. Looks very similar to the Umap cell canasters as the reference data. The query reduction will be reference PCA, then reference reduction will be the PCA, then the reduction mode will be Umap. So let's project the cells to form the cell canasters. So we finished the Umap for the new data. According to the online tutorial, you can just run one function map query. This function included 3 steps of analysis: transfer data, integrate, embedding, and project Umap. It is easier just to run the map query function, but it run these 3 functions for the analysis. So for the demonstration, I run these 3 steps to help you understand the analysis. So now we can plot the reference data as plot 1. So we can plot the new data as plot 2, because we are going to group the cells by the predict cell type. So let's have a look at the metadata for the new data. You can see in the metadata, in this column for the reference data, we named the cell type. Here the column name is the predicted cell type. So we can group the cells by predicted cell types for the new data. So let's plot it as plot 2. Then we can visualize the reference data alongside with the new data. Okay, so we have an error again. That's because this windows is too small, the plot windows. So we can increase the window size, but we are going to zoom in. So we have the plot. We can zoom in. You can see on the left hand side are the reference data set. We have 4 immune cell canasters, 2 epithelial cell canasters, 2 endothelial cell canasters, and 1 mesenchymal cell canasters. You can see for the query data, it has a very similar Umap cell canasters as the reference data set. We have 4 immune cell canasters here, 2 epithelial cell canasters, and 2 endothelial cell canasters, and 1 mesenchymal cell canaster. So you can see once you have a reference data set, you can just use the reference data set to map and annotate the new data set. This will save lots of time for the new data analysis, and also it is easier for you just to look at the cell canaster position. Then you know what kind of cell types they are in those cell canasters. So I finished today's demonstration. So I hope my video can help your data analysis. Please subscribe my channel if you haven't subscribed it, and share my videos with your friends. Thank you, and see you in my next video.